Okay, good morning, everyone. As was mentioned, my name is Diana Ramirez. I am a third-year PhD student at the University of Tennessee, and today I'm going to be talking about my dissertation project, which focuses on microbial biotransformation of PFAS. So some of you might be familiar with PFAS, per and polyfluoroalkyl substances. And so PFAS are known as these forever chemicals, and they are known for their desirable properties of being water, oil, and stain repellent. So we can find them basically anywhere from electronics, cosmetics, clothes, in anything that's water and stain repellent. And recently there's been a study that identifies PFAS in rainwater. So from the consumer products of PFAS through their use and eventual disposal, PFAS end up in wastewater treatment plants, landfills, in our waterways, and through that they are emitted into farmland and food products. And high concentrations of PFAS can lead to detrimental health effects such as cancer, infertility, and thyroid hormone disruption. And currently, there's been a lot of studies on PFAS biodegradation, although there are current mechanisms that allow for PFAS treatment through abiotic methods. A lot of them are inefficient or very costly. And the thing that makes the PFAS biodegradation difficult is if we look at the structure of a PFAS, which is essentially this alkane chain that is completely fluorinated would be a perfluorinated PFAS. And if at least one carbon is fluorinated, then it's a polyfluorinated PFAS. And if we think of chemistry, the carbon-fluorine bond is one of the hardest ones to break down. So just identifying organisms that can do this is very difficult. So in our lab, we study this organism, Pseudomonas species strain 273. And this organism has been able to use mono and bisfluorinated alkanes as a sole carbon and energy source. And so we think of an alkane chain where the first and or the last carbon is fluorinated, and this organism essentially consumes it. And when it breaks down these fluorinated alkanes, fluorine is released into the environment as fluoride, and then that fluoride is taken into essentially the cellular membrane, and we see organofluorine incorporated into glycerol-phosphofatty acids. So we're essentially left with a fluorinated bacterium, which is really interesting. So to learn a little more about this unique phenomenon, we sequenced the genome, and we found a lot of alkane sulfate monooxygenase genes. And so these alkane sulfonate monooxygenase system is implicated in sulfur acquisition from alkane sulfonates. So if there is bacteria that possess these enzymes, if there is a low sulfur concentration in the environment, then microbes can utilize sulfur from alkane sulfonates. And so in Pseudomonas, for example, most of them contain 2 to 3 SSUD genes. However, in our bacterium, we see 22 of these SSUD genes. And so we thought that maybe the abundance of genes would specify for a more diverse set of sulfur acquisition from alkane sulfonates. So this led us to our hypothesis, which is essentially asking the question, if strain 273 can utilize sulfur from per and polyfluoroalkane sulfonates to meet its nutritional requirements. So can strain 273 use sulfur from sulfur-containing PFAS to grow? And so normally in wild-type conditions, strain 273 grows whenever there's no sulfur limitation. However, what I sought out to do was create a sulfur-free medium that would allow for the bacteria to not grow at all since there's no sulfur. And then after it is sulfur-starved, we introduce a type of sulfur into it, and we see that sulfur quickly rises up. And to do this in a more efficient way, what we did is we developed a high throughput testing system where essentially in these wells of this 96 titer plate, we have small sulfur-starved cultures of strain 273, and we're able to test multiple different PFAS at the same time. And then we stick them in a plate reader, which is basically an incubator for a few hours. And then after incubation, we're left with a series of growth curves. And so if we see a flat line, we can infer that that is our negative control. So no sulfur, nothing grows. Or it could be that the strain isn't able to acquire sulfur from that sulfur-containing PFAS. And so to start, I tested a variety of PFAS, so anywhere from things that are perfluorinated to telomeres that are not completely fluorinated, and put them in that high throughput testing system and ended with the following. So essentially, if we see a positive sign or an orange, it means that the OD increased for more than 0.1, which means that we had a pretty big growth curve. And if it's a minus or white, that means that it was a flat line, so probably no growth. And because we can't necessarily correlate an increase in OD with an increase in cell density, for these results, I want to focus on just this type of PFAS, this 4,2-FTS. So after using this high throughput testing system, we made these microcosms. And essentially what we did is we looked at it under the microscope, and we see that whenever there's no sulfur, there's not a lot of cells present. But whenever we have a sulfur source, whether it's sodium sulfate or that sulfur-containing PFAS, there is an increase in cell density. But just to take it one step further after that, we wanted to conduct a mass spectrometry experiment where essentially we set up the following conditions: an abiotic control where we don't have the bacterium but everything in the matrix present, a kill control where we have dead bacteria to see if a disappearance in PFAS might just be the PFAS sticking to bacterial cells, and then 10 micromolar of the PFAS and 100 micromolar of the PFAS. And so what we're hoping to see is that after a 120-hour incubation, we would see that the PFAS would disappear because it is being utilized as a sulfur source, because we already know that without that PFAS, the bacteria will not grow. So after incubation, we see the following. So in our abiotic and our kill control, we see a very low PFAS disappearance, which is pretty negligible. However, and that might be due to the PFAS sticking to the vessel or to our cells. And in our 10 micromolar and our 100 micromolar of PFAS, we see that the 10 micromolar is completely nearly gone, whereas out of the 100 micromolar, only about 30 micromolar of PFAS is used. And that might be because sulfur isn't a complete macronutrient that is needed for cell density. So it could be that 30 micromolars of PFAS is sufficient to make the cell grow. And so what these results tell us is that, well, first, we know that sulfur is necessary for synthesis of amino acids in macromolecules. And so this bacteria is able to acquire sulfur from sulfur-containing PFAS. And from there, it's able to essentially multiply and grow. And after this, what we've been doing is looking at transformation products. So of that list of PFAS that I showed you, we focused on mainly the telomeres because literature has demonstrated potential transformation products. And so what we're looking for are these series of transformation products from different types of PFAS. And we know that this type of reaction can allow for defluorination. And we know that our bacterium can incorporate fluorine into its glycerol phospholipids. So we're essentially looking for PFAS transformation products and fluorine in the cellular biomass. And essentially what our plan is next is we want to specifically quantify those biodegradation and transformation products. And further, we are looking for those products in glycerol phospholipids, whether that's through metabolites, lipidomics, as well as proteomics. And we had seen that there are 22 SSUD genes present, but we're wanting to know which one is responsive that allows for that sulfur acquisition from sulfur-containing PFAS. That way we can develop biomarkers to monitor natural attenuation of this transformation in PFAS-contaminated sites. And all this work is possible thanks to my advisors, Dr. Frank Löffler and Dr. Bob Hettich, and my colleagues in the labs. And with that, I'll take any questions. Thank you.